Correct. Correct. It's not a thermodynamic irreversibility. Okay. So what about these enzymes then? So we've got ketolases and aldolases. Right? Aldolases we already know about because it's the same as the aldolase in glycolysis. So this is an example of a trans-aldolase reaction. This is one of the reactions in that non-oxidative phase that we just looked at. Aldolase enzyme, transfer of 3-carbon unit. How does it work? Shift-based lysine stabilizes the carbanion like the aldolase in glycolysis. So we know something about it. We know enough about it. We don't need to talk more about it. The ketolase, however, is a transfer of a 2-carbon group, and this implicates the cofactor thymine pyrophosphate. And that one we hit before, without talking about the details, when we hit pyruvate decarboxylase. And pyruvate decarboxylase was a step in the fermentation pathway for pyruvate in yeast, which leads to acetaldehyde and then ultimately to ethanol. That's where we encountered it before. It was just one slide and it had TPP on it. So transketolase, this is an example of a transketolase reaction in that pathway. Right? And so one thing to remember about it is that it's a transfer of 2 carbons, and the other thing to remember about it is that it works through thymine pyrophosphate. And now we should look at how thymine pyrophosphate works. Okay, so here are some examples of TPP-dependent reactions. You can see transketolase, pentose phosphate pathway here, ethanol fermentation here. Those are the examples we already hit. Pyruvate dehydrogenase we're about to talk about soon. Okay, there's another enzyme in the citric acid cycle that we'll talk about next time. Okay. And then here we see something called carbon assimilation reactions. And that happens in plants, and this is how plants fix CO2. Okay? So we will talk about that right at the end of the class. Okay? So it's good to keep your mind partly on this stuff as we go through, because we're going to hit it all again right at the end when we talk about light-driven synthesis of ATP coupled to fixation of carbon from the air in the form of CO2 into organic molecules, which of course is what plants do. So here's transketolase and here's transaldolase, and so the resonance stabilization here through the lysine, remember the shift base intermediate here, resonance stabilized, N plus C, C minus. Right? If you follow these carbons down, pretend this is C1 that used to be glucose, 2, 3, the bond just got cut, and we got the carbanion on it right after aldolase cut. And glyceraldehyde 3-phosphate is already dissociated here. I'm just kind of projecting back to what we learned before. And so we had that carbanion, right? And so we know what happens. It's that this guy, the electrons go shooting right back into that nitrogen, right? So this is an electron sink in aldolase. Okay. So here is the business end of thymine pyrophosphate. This is called the thiazolium ring. You don't have to be able to draw it, but you should remember that the stabilization of a carbanion here, C minus, C N plus, C minus, C N plus, right? So it's exactly the same. The only thing that's different is this is on a cofactor, which is a larger molecule, not covalently bound to the enzyme. Whereas here, it is covalently bound just through this lysine link. But so there's 2 ways to do it chemically, but you end up in the same place. You still end up with N plus C, C minus way to stabilize the carbanion by shooting those electrons into that electron sink of the N plus. Okay? Questions? So this is what it looks like, the whole molecule. And here's the business end. So the rest of thymine pyrophosphate is this ring here. So thymine, by the way, is a B vitamin, one of the B vitamins. And then it's got this pyrophosphate link on it. And this is the thiazolium, and this is the thiazolium carrying a 2-carbon group. So when pyruvate, the last step in glycolysis, undergoes its next reaction is going to be to split off CO2, and then the two remaining carbons are going to be doing this. And that's the first step in getting from pyruvate to acetyl-CoA, okay, which is the entry into the carboxylic acid cycle, tricarboxylic acid cycle. Okay, so how does this work? So here's a mechanism. Okay, we will not really look at this in extreme detail, but it kind of goes through some of the same ideas. So here we've got the thiazolium on TPP, right? And the first step is an ionization of the CH bond, not an easy thing to do. Right? So the enzyme is helping bring this about to deprotonate this carbon to generate a carbanion. And the immediate carbanion that's formed here is not the one that we've been talking about. It's a different one. It's on this ring, and it's right next to the N+. Remember, we had C minus C N+. Now we've got C minus N+ directly. So that's not the stabilization of the substrate reaction that we were talking about. It's merely setting things up. The way it sets things up is by generating this carbanion, it generates a very potent nucleophile that can go in and attack some substrate. Right? In this case, the reaction is being shown for pyruvate going to acetaldehyde, which is the yeast fermentation reaction. But the same basic idea in the mechanism would hold in transketolase in the pentose phosphate pathway. So any ketolase enzyme that uses TPP, or any TPP using enzyme that's splitting off a CO2 and carrying a 2-carbon unit is going to work this way. Okay. So this attack here, this attack of the carbanion on pyruvate, generates this transient structure here. Right? We might call this an intermediate. I don't know how long it lasts. And then this is the decarboxylation reaction. So here, then, you can imagine this negative charge starting to build up here on this carbon and then shooting into that nitrogen. Right? So this is the analogy right here to what we learned before. Okay? So this is the CO2 leaving now. Right? CO2 is decarboxylating. This bond is breaking, right? But unlike in aldolase, where we had a 3-carbon compound on the other side, all we get on the other side here is CO2. So goodbye CO2 and carbon fully oxidized. Not like 6 carbons going to 3 plus 3, but in this case, 3 carbons going to 2 plus 1. The 2 remains stuck on the cofactor, and the 1 is gone, shooting off as CO2. Questions? So we'll skip the second part of that. Okay, so again, not a mechanism that you need to be able to write out from scratch, just one that you should be able to answer questions about. Okay.
Actually,
Yeah.
I mean, I'm not trying to get test answers here, but what kind of things should we be able to answer about this?
Well, you should know what common features are present in a thiazolium ion and a protonated shift base, for example. That kind of stuff. If I tell you that the carbanion is generated on TPP and show you the structure, and I tell you it attacks pyruvate, and you know what has to form, then you might need to remember where on pyruvate it attacks. But you can remember that because you know what needs to form. So stuff like that.
Okay.
Okay. So pentose phosphate pathway is another, it's kind of like a bypass, right? It's a way to make NADPH, it's a way to make ribose sugars for nucleic acid synthesis. We recycle carbons. We start to get a feeling for, how all these sugar reactions are interacting with each other. Because now we've seen the feedback.